Good evening, all. Let's get started with today's live session. So, this will be the seventh live practice session for the course Applied Environmental Microbiology, and I am Guru Priya, Prime Minister Research Fellow in the Department of Civil Engineering, IIT Madras. I hope all of you have gone through the lecture videos of week 7 and all the previous weeks before. So, the contents include techniques in environmental microbiology and bioremediation. Uh, you can answer this, try answering this question. We'll get started with this. And also, if you have any doubts from this week's session, you can ask at any point of time. You can put it in the chat box or unmute yourself. You can ask it at any point of time. I'll try clearing up your doubts. This is the first question for this week. I hope all of you remember what a Vinogradsky column is. I just ask like where will algae be found in which zone? There is no particular naming of a zone. I'm just asking the position of the zone to be clear. Okay, one person has responded. I'll wait for a few more to think and answer. There is only one response. How about others? What happened? Are you thinking about what a Vinogradsky column is? So one person has told its top zone. What do others think? Will it be in the top or bottom or in the middle? Which zone do you think algae can be found in a Vinogradsky column? Okay, with that one response, let me just show the answer. It's right. It will be present in the top. Um, Okay, for what does a Vinogradsky's column is used? Can anyone answer? For what is a Vinogradsky column used in environmental microbiology? Why do we need a Vinogradsky column? Okay, that is one answer. Anyone else would want to give a try? Okay, so as shown in this picture, Vinogradsky's column is generally used to culture multiple microorganisms. Okay, large diversity of microorganisms can be cultured in a Vinogradsky's column. Uh, the basic principle is that you're going to, as Nidhi Praveen has told, you have to mimic the lake. So mimicking in the lake in the sense, we will add mud, we will add water. So this entire slurry will be something that mimics the lake ecosystem, basically. So what happens is when you are going to see what is the diversity of microorganisms that is present, we will find different kinds of organisms in different layers. We have also talked about stratified lake and all in the previous live sessions also, and in the lecture also professor has discussed about all of this, right? So what happens is we have a column, we put in this water, mud and all, and then we cover it so that we have a closed system. What happens is then this zone separation, there is no very distinct difference, like you can't visually, it is not very appealing. Just by the color you have to differentiate. You can see aerobic zone at the top because it's in contact with the air zone. Here you will have some space here, so this will be containing air. You're not creating any vacuum or something here, it's just air. So this layer is in direct contact with air, so the dissolved oxygen or the oxygen transfer is more. Whereas when you go down, this oxygen transfer will automatically get lower. Also you have all microorganisms here, so what happens is they tend to align themselves to the to respective zones based on the metabolism that they can carry out. So at the top where you have this aerobic zone, you can find cyanobacteria, algae, all these aerobic organisms. And then this micro aerophilic zone. What are micro aerophiles? Can anyone let me know in the chat box? We have discussed this earlier. Maybe in the first few weeks, 2 3 weeks, we have discussed when we talked about classification of microorganisms. We know aerobic microorganisms require oxygen for survival. What are microaerophils? I mean, right or wrong, you just try to answer. This session is basically to uh, enhance your capabilities to answer or face the questions. That's it. It's not like you have to tell the right answers. And just give a try about what are microaerophilic organisms. Okay, if microaerophilic is also aerobic, then what's the difference between both? We have separate classifications, right? Here it's aerobic, here it's microaerophilic. If microaerophilic is can be completely accepted as an aerobic organism, then why do we have a different classification of microaerophiles? There is some significance, right? Okay, let me tell you. See, whatever name it is, first of all, try to understand it from by splitting the name. Okay, even if you are like not aware or anywhere when you're not even if you're not aware, just try to see what part of it you know. So this is a very basic information. So coming back to the content here, micro aerophiles you can separate as micro, air, and philic, right? So as they are aerophilic, they will require air, okay, but they are not, they cannot survive in normal oxygen tension and concentration, okay. Uh, that's not what they can easily metabolize. So they like environments where the air is minimum, like a low concentration of oxygen is what they like micro air, so that is why they are micro aerophilic. Okay, is that clear? Microaerophilic organisms also require oxygen, but they don't require oxygen in such a concentration as the aerobic organisms. They can survive in conditions where the oxygen concentration is a bit lower. Then what's the difference between anaerobic and microaerophilic is they cannot survive in the absence of oxygen also. They cannot survive in normal oxygen concentration, like we say 21% of air is oxygen. We have such classifications, right? So in that conditions also they cannot survive. They cannot survive in a complete oxygen deprivation also. Like when oxygen concentration is zero, then also they cannot survive. So that is basically the difference, that what differentiates microaerophiles from aerobic and anaerobic. That is why we have a separate layer also here. I think this picture is very vaguely visible, but still you can try seeing it. When I upload it in the drive, you can see it a bit clearly, I guess. So in the aerobic zone you have, and the microaerophilic zone you have. uh, the microaerophilic zone usually contains this purple non-sulfur photoheterotrophs. uh, so and they have a rusted color or a red brown color, okay. And then comes the anaerobic zone. So in the anaerobic zone, you can find purple sulfur bacteria, and then the green sulfur bacteria, and then the sulfate reducers. So this reaction is the reactions are basically representing what is the electron donor, what is the electron acceptor, and like what happens in their metabolism. That it's just an overview of that. So as told in the Vinogradsky's column, what we do is we mimic the environment such that the top layer is in contact with air. So aerobic organisms will get a good amount of oxygen in that place, and they will be in the top. Aerobic organisms include algae, cyanobacteria, etc. So that's what I asked. Where will the algae be? Algae will be in the top layer. As when you move down, the oxygen concentration is going to decrease from top to bottom. So next you will have the microaerophiles, followed by anaerobes. Is that clear about Vinogradsky's column? Any doubt? Okay. Okay, other people, it's just like more than 10 minutes since we have started this session. At least give me a thumbs up now. We can move to the next slide. Okay. Okay, at least one person responded. So this is your second question. This is a true or false question. Okay. As we have followed till now, when it is a statement, it's just asking true or false. When there is a chance of product inhibition, sequential batch reactor is preferred. Is this statement true or false? I think more people can answer for this question because we will see how much people support true, how much people support false. Okay, let me tell you. If you're going to answer true or false, I'll not ask the reason for that and then I'll explain the answer. Is that okay? Can I get more responses now at least? You can just give true or false, whatever you, based on whatever logic you think. Okay, I see a couple of thumbs up there. I'll wait for these people to think and answer in the chat box. Okay. Okay, so now let me ask you, how many of you have no idea about a sequential batch reactor? You can just give a thumbs up, that's fine. Just give a thumbs up for a reaction so that I know that you don't know about a sequential batch reactor. Okay, that's good. You still tried answering this question. Okay, seems like I did not get much of a thumbs up, so you people know what a sequential batch reactor is. I guess someone has raised your hand. I cannot see the name. If you have any query, you can unmute or you can put it in the chat box. Okay, so we can, I'll first tell you what a sequential batch reaction is. So I guess you all know about what is a batch, fed batch, and a continuous reactor, right? A batch reactor is a reactor in which you're going to add your substrate, the inoculum, and leave it aside. Your reaction will take place. Maybe say your reaction time is going to be 2 hours, and then after 2 hours you come back, you will have more biomass in the reactor, obviously there will be growth, there will be production of some product, utilization of the substrate, and production of some product. Okay, so you're going to take it back and separate this micro product and all based on your interest, and go for further processing of whichever product you need. Okay, this is a batch process. In a fed batch process, what you do is you add your substrate inoculum, allow it to react, and in between you again add more substrate. So first you add some substrate, and until certain time you will be adding more substrate, but you will not be removing anything from the system. You are just going to add substrate to the system. So that's fed batch. And in the continuous mode, what happens is there will be continuous input of the substrate and continuous output of your product. You will be putting in the substrate, taking out the product. So this is, I think you are clear with what is a batch, fed batch, and a continuous system. Okay. So here, when it is a sequential batch reactor, again you have to split it word wise. So it's a batch reactor. Obviously, you're going to put in the contents, allow them to react, and then only take the product out of it. You're not going to disturb it anyways. No inflow or outflow during that reaction time period. Right. And in as the name suggests, in the sequential you're going to have few tanks or the reactors connected in a sequence. This is not something like a reactor in series or something. If someone has studied maybe I think in reactions engineering and all, you'll be studying about all the series reactors in series and all. Please don't confuse with that. It's just you're going to arrange few tanks in a sequential manner. Okay. Generally we use this SBR in wastewater treatment, where you will be having them in different stages. Like you have to fill it with the wastewater, you have to aerate it, you have to allow it to settle, you have to remove the sludge from it. So all these happens. Basically it has a batch reactor characteristics. When I say product inhibition, it means the product formed itself will inhibit the microorganism. So that is one case. Okay. I think if you have studied about enzyme kinetics, even if you have not studied, that's not a problem, but if you have studied about enzyme kinetics, you would have heard about many kinds of inhibitions. Inhibition is a general term used. Okay. Inhibition means something is hindering or stopping the activity of an biomolecule or a microorganism. Okay. In this question, what I have asked is about product inhibition. That means in a batch reactor, in a sequential batch reactor, a reaction is happening and some product is formed as the by the utilization of substrate by the microorganism. Okay. Above some concentration, this product itself will be inhibitory to the microorganism. Let's say I get ethanol as my product. Okay. After some concentration, this ethanol itself will become toxic to the microorganism and it will not be surviving. So this phenomenon is called product inhibition. I guess the question is clear now. Okay. I'm telling that when there is a chance of product inhibition, I will be preferring only sequential batch reaction, and I'm asking whether it's true or false. So okay, with all this explanation taken, would anyone like to answer this question now? Will it be true when there is going to be product inhibition? Will I go for a sequential batch reactor? I have told you in a batch reactor, I'm not going to take out any of my products, or I'm not going to put in any of the substrate. I'll just wait for one more minute. Okay, so as two people have answered, it's false. Why false? Because mine is, as I told, is a batch reactor, so I will not be removing any of my product, and all my product is going to stay in my reactor only. If my product is going to stay in my reactor beyond a particular concentration, it's going to inhibit my microorganism, and the process is going to be hindered. I will not get the efficiency or yield that I thought I would, or that I expected to get. Okay, so that's why when there is a chance of product inhibition, sequential batch reactor will not be preferred. Okay, I guess I have given enough explanation. If you want any more clarification or repeat, you can let me know in the chat box. I'll just post the third question. Okay, here we have two answers, one false and one true. Anyone else would like to try? We are in a tie. We have one false and one true. Okay. Okay, so the answer here is true. Okay, so this is the graph. I think you have seen this in the lecture. S- Basically, in microbiology or here, R strategists and K strategists is just depending on the acclimatization of microorganisms. So, K strategists are able to adapt to a crowd and a resource limited environment. Okay, so K strategists can survive in a little lower um, what to say nutrient concentration and a higher competition of microorganisms basically. So here what happens is, so you can see when you are having a substrate and are adding a microorganism or a group of microorganisms to the substrate, there will be substrate consumption, so your substrate is going to come down. Okay, and then you are going to transfer it to a new medium containing fresh nutrients and substrate. So instantaneously this value is going to raise, the substrate is again going to go, let's say this is 0, this is 1. So yeah, when I start with 1, it eventually goes to 0, and suddenly when I'm going to transfer, it again goes to 1, and the same pattern is going to follow. So here at one point of time we will find a group of microorganisms called R strategists, which are not that much competent to grow in a crowded environment or a resource limited environment. They just need enough food to survive, they just need a less congested space to survive. And there are K strategists which can grow in a crowded environment, which can grow, they just don't require so much of food, they can survive with a little food. Okay, so I'm asking, I'm telling that the case strategist sampling should be before transfer to a new batch media. So why is the answer true? Because at this zone, when there is less crowd of microorganisms and more availability of substrate, you can find the R strategists. Eventually this goes down, and here you can, here the resource gets limited. I think you can see the pointer here. So at this zone down, your resource is getting limited, and the microorganism count will be increasing as they consume the substrate. What they do, they are going to multiply, the cell is going to multiply and you get more biomass, so that becomes a crowded environment again. So in this resource limited and crowded environment, your K strategists will be more dominant. You can see more of K strategists there. So if you want to know what are the K strategists that are present, you have to sample just before transferring it to a new media. If you want to know the R strategists, you just have to sample just right after transferring it to a new medium. So in this zone you will have R strategists, and this zone you will have K strategists. So for R strategists study, you have to sample it just right after this transfer event, and for case strategies you have to sample just right before the transfer event. Is that clear? This is basically to understand the microbial dynamics or the community dynamics. Okay, okay, so I think I've got a couple of responses for it. If you want me to repeat, just put it in the chat box so that I can repeat. So for now, I'll just go to the fourth question. I'm asking about the oxygen concentration and the electron donor for a green cell for bacteria. Okay, there is a handful of responses, and yeah, all of you are right. Um, Leakethite is not aerobic; it's anoxic. Like the green sulfur bacteria cannot survive in the presence of oxygen. Like, so the green sulfur bacteria, as the name indicates, is a sulfur associated bacteria. So the electron donor will be hydrogen sulfide. It can metabolize hydrogen sulfide, and it does that in the anoxic condition. So obviously using oxygen is very easy for most of the microorganisms. This happens in anoxic conditions. So with that, like I think I explained it before also. What's the difference between anoxic and anaerobic in terms of metabolism? They almost mean the similar things, but if someone wants to answer it by unmuting, you can do, or you can just give hints of your answer in the chat box. So we use two different terms, anoxic and anaerobic, right? What's the difference between them? I'd also ask you one more question. What are all, based on what factors, like we have studied the classification of this microorganisms, uh, in terms of this nutrients and condition is based? I remember in one week, week 2 or 3, I had a flow chart explaining different classification of microorganism based on this mode of nutrition that it has. Anyone? I think we have a lot of questions to go, but are you understanding the question? I just asked two questions. One was, we have anoxic and anaerobic, two different things. What's the difference? Why do we have two different concepts called anoxic system and aerobic system? Another was, what's the classification criteria? Like here one is present, like electron donor is one of it. Like that we had few more criteria. Okay, so you people know the answer or you want me to explain? Anaerobic oxidation. Okay, one person has told me to explain. I'll wait for few more responses. If you people know, you can type the answers. One person has already answered. Given one answer. Anoxic is anaerobic oxidation. Okay, so let me explain it quickly. Second question first. Classification of microorganisms. So generally, we classify microorganisms in terms of this growth and nutrition context as based on their energy source carbon source, electron donor, and electron acceptor. Okay, energy source, based on energy source, what are the sources? Quickly, it's light or it's some chemical substance. So either they are going to be phototrophs or they are going to be chemotrophs. And again, based on carbon source, either they will need organic carbon source like we humans do. We need organic carbon source. We just can't take carbon dioxide, metabolize that, get our energy and survive. Okay, so it's going to be organotrophs when they are organic carbon users, so or heterotrophs as you can call, when they are going to use this inorganic carbon such that carbonates, carbon dioxide and all, we call them autotrophs. Okay, so two things we have seen: energy source will give you either phototrophs or chemotrophs. Carbon source will tell you whether it's a heterotroph or autotroph. Again, electron donor, based on electron donor, the donor can be organic or inorganic. Okay, when it is organic you call them organotrophs, and when it is inorganics you call them lithotrophs. Okay, and the electron acceptor you have, that is what is the key difference in this aerobic, anoxic and anaerobic system. Okay, the electron acceptor can either be oxygen or inorganic substance or organic matter. You can have three. When the electron acceptor is oxygen, we call it aerobic respiration. Okay, it's called aerobic respiration. When it is inorganic substance, we are going to call it as anoxic or anaerobic respiration. Okay, and in the third case, when there is organic matter, we call it anaerobic fermentation. So that's when the term fermentation enters the context. Okay, first you have oxygen, which is the electron acceptor. That's the most easiest way. I guess you all remember about the electron transport system. Okay, the electron transport system, I think you have to go back to all your basics now if you are not understanding this concept. So please listen carefully. So the electron transport system basically functions based on the electromotive potential. Okay, so that's how the electron transfer happens because of this potential gradient, and because of that only ATP is generated. Okay, so that's a very key step in the microbial metabolism. So in the electron transport chain, uh, how is this potential created? We have seen a redox tower, right? Where we can see electron transfer from top to bottom of the tower, right? So in that case, when the final terminal electron acceptor is going to be oxygen, you will have a very good or a complete electron transport system. So the ATP production will be more, and that's basically the aerobic system. Whereas in the case of anoxic system, as Shagupta has rightly mentioned, it's going to be oxidation only, but it will not go until oxygen. We have seen that when you are going until oxygen, oxygen will be the lowest. So when you are going until it in your redox tower, you will have a greater potential, electromotive potential. So the energy that is released in the form of ATP will be higher. In anoxic, what happens is you will have either sulfur, nitrogen, such things will come into the picture, which will have a greater potential. So it will stand above oxygen in the tower. So the electron transport system is going to be shorter. So from here, for oxygen it has to travel this much. If it has to travel this much, for other source like the inorganic source is not going to travel that much. It's just going to stop in halfway. So it will not produce that much of ATP or that much of energy. So this is known as anoxic respiration. But still you have this oxidative phosphorylation only in that case also. But in the case of organic matter, what happens is mostly you can see this concept of substrate level phosphorylation, and it's going to be fermentation. So organic matter, when you are going to when it's going to act as electron acceptor, what it will do, it will form products such as lactic acid, ethanol, Okay, so when it is organic matter, it's going to be fermentation. Ferment, fermentative metabolism is going to happen. So in this case, green sulfur bacteria has an anoxic condition with hydrogen sulfide as electron donor. Electron acceptor will also be some other inorganic substance only. Okay, is that clear? The difference between aerobic, anoxic and anaerobic systems. Is it clear or you want me to explain again? Okay, anyone else would want me to explain again? Because she gave the right answer right before. Oh, sorry, I did not notice if you gave a thumbs up or something. If it's clear, can you just give me a thumbs up? We'll just move on. Right, I just got one, so with that I'll just go. Okay, we have, okay, we have different sets of answer. 1, 3, 1 and 2. I guess most of you are clear about it's 1 from those who have answered, because okay, finally one person has come to the conclusion. With all of this, it's all of the above. So I'm not asking about whether they can whether nitrogen or nitrogen related compounds are used as electron donor or acceptor. That is where this concept of all this nitrification, nitrogen utilization and all comes. It's just a straightforward question whether nitrogen is used as a nitrogen source for enriching these bacteria. So we generally I don't know if other people than microbiologists or biotechnologists know. Um, when we do prepare a media, we just see what is the carbon source, what is the nitrogen source, because carbon and nitrogen are very important for the cell. So we just see when you're going to do a pure culture, you're just going to prepare some medium and all. You're going to prepare the media yourself and then do a pure culture in that. You just go for a very detailed calculation of what will be your carbon source, what will be your nitrogen source, how much you will add, what's the ratio of all of it, what will be the trace elements that you will add, because all of the requirements of the microorganism you have to satisfy, so you will go for it. Here I have specifically mentioned what is the nitrogen source. Nitrogen is the nitrogen source. Obviously nitrogen can be used as a nitrogen source for enriching all of this bacteria. Okay. So next question is, for enriching dark chemolithotropic bacteria, the medium must lack what? Dark chemolithotropes in the sense they require dark conditions, they do not need light. Other than that, this chemolithotropic, you can just rewind or recollect what I had told few minutes back. Okay, so as you have all told, it should lack organic carbon. So chemolithotropic metabolism does not support this organic carbon presence. So you just have to have that flowchart in your mind about this carbon source, energy source, electron donor, and electron acceptor. In any such question, you can just logically think and answer, even if you don't know exactly about the organism and all. Another similar question, just for practice. Electron donor for a thiobacillus species include. Okay, so there are multiple answers and all of it is right. The electron donor for a thiobacillus. Thio, usually we use it for sulfur sulfur related things. Okay, that is the catch here. So green sulfur bacteria, purple sulfur bacteria, you will know that it's going to be associated with sulfur. Non-sulfur bacteria also, you just have to know how to interpret the name. So even you when you can't learn all of the concepts. Okay, uh, you just have to know how to integrate the name, what specific things mean, the basic fundamentals like carbon source, energy source, what do they mean, what is their significance and all, so that you can answer any such questions. Like most of the questions you can answer apart from very specific questions and all. So in this case thiobacillus means it deals with something sulfur. So the electron donor can be, as we discussed in the last slide also, it's going to be hydrogen sulfide or sulfur with zero oxidation state or sulfite. Okay, right. This is a very technical question. Like, I mean, those whoever has performed this will know. I just wanted to let you know about this concept, so I have included this question in the slide. So all this while we have been, yeah, many answers of so enthusiastic about autoclaving, right? So all this while we have been talking about plating. Okay, right from the beginning, right from maybe week one, we have started talking about this plating technique. So the plating technique basically requires a solid medium. So what do we use as the solid medium? Agar, right? So those who don't know about what an agar is a polymer. Okay, this polymer, once you heat it and then cool it, it's going to get solidified. That's very basic. Okay, uh, I think I have also told you about agarose gel and all. They are similar. Like all of these are polymer. The tendency is when you're going to heat a solution of agar and then let it cool, it will solidify. I think all of you have eaten jelly, right? In homes, what do we use? We use maybe if you see the packet of this jelly powder in which you can prepare in the home and all, it's going to contain some polymer like this. Most probably it's agar, so that when you're going to boil it and then when you're going to allow it to settle, it'll come to that jelly consistency. So that's how your medium is also going to look or be like. Okay, so this medium has to be first autoclaved, because the microorganisms in the environment itself can eat all the substrate that you have provided, right? It's not guaranteed. It's not like I have provided the media only for the microorganism that I intend to grow, so only that should grow. It's not like that, right? Whatever microorganisms is present in the environment can grow in that plate. That we don't want. So what we do is we prepare this nutrient agar. It's just, maybe you can get it like in a packet or box also. You just have to add a particular amount of that agar, nutrient agar, into your water, distilled water or deionized water. You just add it inside it, and you have to do a process called autoclaving. So in autoclaving, what you do is you just apply heat and pressure. Okay, so as everyone has mentioned here, it's 121 degrees Celsius. So this is the first thing that microbiologists or biotechnologists who work with microbiology will be taught. You have to have that 121 degrees Celsius and the 15 psi pressure. I think all of us will have this in mind forever. Okay, so this number, uh, you may have a doubt what is so specific about this number. Will you people also be liking to answer why do we choose 121 degrees Celsius? Those who have answered 121 already. At least one person. Why do we use 121 degrees Celsius? Why can't it be something else, more or less? Right. To kill microorganisms and spores. If you remember, spores are dormant structures. Uh, you cannot kill them so easily. So when you're going to apply 121 degrees Celsius at 15 psi pressure for 15 minutes, so what happens is all the spores, all other microorganisms that is going to be present in that will uh, die. So so that your medium is getting sterilized. Only after that what you do is you allow the pressure to go off, like you do in a normal pressure cooker that you cook rice. What do you do once the rice is cooked? When you get the whistle, you just switch it off and you don't open it all of a sudden, because there will be high pressure. What Because of the water vapor that was generated, you allow it to cool down and the pressure, release the pressure, and then you open it, and then you take this medium to your lab and pour it in a plate, and it will solidify, and then you will plate on it. Okay, this step is very important. So autoclaving is very important, I guess. uh, it's clear now. So we'll see the next question. In a plate count method, the CFU must be between dash for valid results. Okay, as all of you have rightly mentioned, it's 30 to 300. It should not be less than 30 also, it should not be more than 300 also, because if it's going to be more than 300, it will be very tedious to count it, and you will obviously end up getting errors. And when it's very dilute, you cannot extrapolate it. So your dilution should be such that you have to plate that dilution and get colonies between CFU of 30 to 300, and when you're then going to count the colonies, you can have it as a reliable result. All this you have to know when you're going to work with these microorganisms in the laboratory. indicator used in anaerobic culture is while doing anaerobic culturing. When you're going to culture anaerobic strain, okay. So here I have mentioned methylene blue, and we also have a answers of the Rios urine. So both of them are right only. As far as this course is concerned, whatever was discussed based on the lecture, it's methylene blue, because when you have a jar inside this, you can put your plates, and then when you add methylene blue in that jar, when there is going to be oxygen only, methylene blue will be blue in color. When it's going to be anaerobic condition, it will turn colorless. So that will be a very good indicator for us whether the jar in which we have placed the plates has oxygen or not. So when you're going to see a blue tinge or a present, when you're going to see the blue color of the methylene blue, you're going to confirm that okay, the air is present, so it's no more anaerobic, and you're going to troubleshoot. Also, reassurance is a pink color indicator again. That is also used to confirm whether it's in the aerobic state or in the anaerobic state. So you can use both of it, but very basically you use methylene blue. When you're going to extensively work with anaerobic cultures and all, you will obviously get to know about reassurance and many more methods. You have separate anaerobic chambers where it's exclusively has anaerobic environment in it. You can work in that also. So there are many ways of culturing it. Two things can be reassurance as well as methylene. So this is the next question. Any more answers? We have qPCR, we have microscopy. Okay, so all of you are sure about plate count being used for counting microbes. We have seen about CFU, PFU in case of viruses, all stuff. Okay. What is a qPCR? Quantitative PCR. Okay. So, okay. So Parul has answered it was microscopy, and then probably a justification. Quantitative PCR copy number of bacteria is determined in it. Right? Okay. So, as all of you have told, qPCR stands for a quantitative PCR only. It's generally used in a diagnostic purpose, right? With quantitative PCR, you can also count the bacterial load. You can use those amplicons to get the bacterial load. Okay. So qPCR can be used to count microbes. Plate count can also be used to count microbes. Now, one person has answered it's microscopy and no one has told it's serial dilution. Why do we do a serial dilution? To dilute the concentration for ambient test, yeah, to plate with less microbial load, and we use it during plating. Okay, to dilute the concentration during plating. Okay, so serial dilution is a technique in which you're just going to take your sample which has, let's say, 10 to the power of 10 cells, put it somewhere into the next tube. You just add it and it reduces to 10 to the power 9 cells. All you have is an assumption in mind. Okay, it's just you are assuming, you don't know you have 100 cells. You're going to put it in the next tube, it's going to change to 50 cells and then to 25 cells. You don't have a count of it. So, for serial dilution is not a technique where you count microbes. If that is the case, then you will not use plate count. After doing serial dilution, as you people only told, serial dilution is first done, followed by plate counting, right? Followed by plating it. Why do you do a serial dilution and then plate it? Because your plate should have lesser count. Only that plating technique counts in as a micro counting technique, not the serial dilution. Serial dilution is just like a preparatory step. Okay, serial dilution is basically a preparatory step. In that step itself, you don't know, okay, I have 100 microbes when I'm going to put it in the next tube, where I have this fresh sterile water or something, or sterile saline solution or a buffer, it's going to reduce to 50. So this will be the number. You're not going to know that in your serial dilution itself. Only when you're going to plate and see how many colonies are growing, then only you will know what is the count in the initial sample. You can use microscopic techniques for counting microbes. You can just visually see them under a microscope, and that's a very tedious process, yeah, but you still can do it. I think you have this petrophosphor plates and all, which are like grids. So with the help of those grids, you can add some number of samples, and count it, do a particular multiplication kind of a calculation, and then extrapolate it to how much of microbes is present. So qPCR, quantitative PCR, can be used to calculate the microbial load. Plate count, I think all of you by now know that plate count is used for microbial counting. Microscopy can also be used for microbial counting, but serial dilution is not a technique in which you count microbes. Okay, is that clear? I really felt it surprising that no one answered serial dilution for this question. I think it's clear now. Okay, good. So the answer is serial dilution. And next question. What is a DGG? Right. DGG stands for denaturing gradient to electrophoresis. So we have true, true, and false. We'll just wait for a few more responses. Maybe a couple of minutes more. Okay, just a little recap. Electrophoresis is basically a technique where you're going to put your DNA through, in the presence of some potential difference, so that based on the size and charge it will move. So DNA is negatively charged. When I'm going to apply a potential, it will move towards the positive electrode. And for the electrophoresis, I will be using a gel. So that gel will be porous in nature. So based on the size only, the movement of DNA will be there. The DNA, if I have more base pairs, the size is more, it, what happens, it will not travel farther. When it's smaller or lighter, it can travel farther. So when you're going to read the gel from top to bottom, the heavy ones will be at the top and the lower ones will have moved farther and will be at the bottom of the gel. Okay, this is just an outline of electrophoresis, as we have mentioned in the earlier sessions also, gel electrophoresis. um, Here we use this denaturing gradient gel electrophoresis. Denaturing in the sense we are going to denature the DNA. Denaturing, separating of strands is known as denaturing in the case of DNA. We also have a phenomenon of denaturing of proteins wherein the structure gets disrupted. So denaturing is used for different biomolecules. In the case of DNA, the separation of the two strands can be called as denaturing of the DNA. I think in the PCR, when I explained the steps, I told you it's denaturing, annealing, extension, and then renaturing. All this I told you, right? So denaturing means separation of the two strands. Okay. So when you're going to denature, it's basically breaking the hydrogen bonds in the DNA. AT and GC. AT has a double bond, hydrogen bond, whereas GC has a triple bond. So based on this only this question is framed, this simple concept. When GC bonds are more, breaking of the DNA will be hard because it's a triple bond. Comparatively, double bond will be broken easily. So the GC bond breakage is a bit difficult and DNA does not travel farther in a DGG. This statement is false. It will travel farther in a DGE because this is a gradient electrophoresis. Okay, in a normal electrophoresis, also the normal electrophoresis, the heavy molecules will not travel farther, but it's a gradient electrophoresis. When the GC bonds are more DNA does not travel farther because they will be intact, they will be bigger in size and they will not travel farther. Is that fine or is it confusing? Should I repeat? I guess it's a simple concept. Okay, I'll go to the next question. I hope you're all aware what bioremediation is. You're going to remediate with biological methods. How can you accelerate bioremediation? Or what will facilitate this bioremediation process at a faster rate? Okay, most of you have answered bioaugmentation and biostimulation. What is bioaugmentation? Or maybe you can just answer both. What is bioaugmentation? What is stimulation? Or you can just give a difference between both. They are just different terms with different meanings. You have to know what they mean. You can just give one answer, like either bioaugmentation or biostimulation, just in 2 3 words. Okay. When you know what they mean, you know why they act as accelerators of bioremediation. It's simple. Right. Many of you have answered it right. So bioaugmentation, addition of specialized microorganisms. So augmentation is generally enhancing something that's already present. You just augment it. You just add something more so that it becomes more robust. Okay. So by augmentation means adding something similar to enhance it. So keep that in mind. So bioaugmentation means you're going to add microorganisms, more microorganisms that are capable of different metabolism or enhanced metabolism, whatever be it. So this will accelerate bioremediation. And it's going to be biostimulation. You're going to stimulate something. Okay. So for stimulating something, you need a stimulant, right? What will be the stimulant here? It will be the nutrient or the surfactants, electron acceptors, electron donors, or anything that can stimulate the process. So once you stimulate it, it will go at an accelerated rate. Okay. So biostimulation means you are adding some component like a nutrient or electron acceptor or donor, some component. In augmentation, you are going to enhance the existing, that means enhance the existing microorganism only. So in that case, you will add microorganism. This is the difference between augmentation and stimulation. Okay. So with that, we'll go to the next question. When the contaminant is going to be converted to carbon dioxide in water, the process, the complete process is known as. Okay, as Shagupta Khan has rightly answered, it is known as mineralization. So when you're going to see, for example, I have a contaminant, I do some steps, it's not going to go to a carbon dioxide and water state, which is the final, like completely oxidized state. I will not call that process a mineralization. I'll call it as a transformation, remediation, or anything like that, based on what process is happening and what product I get. When I completely convert the contaminant to a carbon dioxide and water, you call it as mineralization. Okay, so the next question. Very easy question. I just told the answer now, few seconds back. This is a continuation to the previous question. Hmm. Okay, these are the basic concepts. So I just saw the answer. It's transformation. You generally call it as a transformation process. One thing is transformed into another thing. And it's not always that I have mentioned that the intermediate metabolite that differs in toxicity. So it's not always that when I convert one existing molecule to another molecule, that difference in toxicity is going to be lesser toxicity. That's not the case. I transform A to B, for example, and I say they differ in toxicity, you tend to inherently understand that B is less toxic than A. I have converted it into a different toxicity means less toxic only, but that's not the case. Sometimes you convert A to B and B tends to be more toxic than A. Even that happens when you do this waste treatment, basically wastewater treatment and all, uh, which we generally call as byproduct formation and stuff. Okay, so that's one key point to remember. So here's the next question. All these are general terms that you have to know with their right meaning. Parul, for the previous question, it's not bioconversion because I have not mentioned that it's in a biological way or something explicitly. This is also a term that was extensively discussed in the lecture. Any compound not occurring within the normal metabolic pathway of biological system. Okay, let me give you a clue then. Let me rephrase this question as any compound that is foreign to a given biological organism is called any compound that is completely foreign to the biological organism is called right. Now you get it right. So you have to be aware of what it means from different perspectives. Okay, when it's completely foreign, what do we mean? The organism that not produce it at any of its metabolism. That's how a product can be completely foreign, right? This is one way of telling it in terms of toxicology and stuff. So okay, we'll go for the next question. It, just few more basic questions left, and with that we'll close the session. I think this also during bacterial growth and all we discussed. When there are more than one carbon source, dash growth is seen. So yes, the answer is dioxy growth only. Dioxy growth in a sense it will have two lag phase. For example, in dioxy growth, what happens is you have two. For example, let's say we have give two carbon sources. One is glucose and one is lactose maybe. Okay, so lactose is not very easily metabolized. Like glucose is more better metabolized by the microorganism or more easily metabolized by the microorganism than lactose. So first what happens is you get a lag phase and then a log phase that constitutes the consumption of glucose, and after that it should ideally go to a stationary phase, but here since we have one more carbon source, what happens is again it get used to this lactose, so it goes into one more lag phase and then goes to another log phase with respect to lactose consumption, and then it goes to stationary phase in the decline phase. So dioxy growth can be seen when there are more than one carbon sources where you have this two lag phase and two log phase. Okay, the next question. It's just a variant of the true or false question. I think it looks voluminous, but it's a pretty easy question. Okay, so aromatic compounds are generally those cyclic ring structures. They are not easily degraded by microorganisms. It's tough to degrade them because of their structural rigidity. And also, physical methods and all will not effectively remediate aromatic compounds. You will require a lot of energy then to break the bonds and all. Okay, so it's not very effective. Aromatic compounds can be remediated only by chemical oxidation. No, we can use other methods also. The effectiveness varies, that's for sure, but you can use physical, chemical, biological methods. Okay, the effect, if the options are interchanged, then that's a case like aromatic compounds effectively degrade by chemical oxidation, then you have to think about the mechanism, effectiveness and all, and then you can come to a conclusion. But it's not that only chemical oxidation method can remediate aromatic compounds. So it's that aromatic compounds are generally challenging to remediate due to their stability and toxicity. I just have one more slide, I guess. So this one and next one. So we'll close with that. Okay, so the answer for this question will be D only. Acclimation is a process by which organisms adapt to the changing environmental condition. It's a slow process. They cannot like rapidly you put it into some different environment and just get adapt. It's not the case. You see, right? Even in microbes, you have this lag phase. So they take their own time, so it's not a very rapid process. Like you put it there and it starts like adapting and growing well. And then it not only occurs in plants and animals, it not only occurs in plants, it also occurs in animals. Even we can get acclimatized. And also acclimation is not a permanent genetic change. It's not like inherited and all. Sometimes it's just for that single organism only. Like we saw a concept about epigenetics and all. Like your environment uh, turns on or turns off your gene. Sometimes that kind of a change can take place. Sometimes it will not take place. Sometimes the genetic change can be inherited. Uh, in the epigenetics, it's just mere switching on or off of genes, right? So it can be either of it. The last question for today. I'll just wait for only 1 minute and then we'll close. Okay, I'll quickly go through. The answer for this question. So, PHB, as you all know, is polyhydroxybutyrate, which is a bioplastic. The degradation is an important process for remediation of plastic waste. Okay. PHB can be degraded by chemical or other methods also, thermal methods also. It's not like only bacteria. Uh, Similarly, bacteria can degrade PHB, so obviously enzymes will be used, and that statement, C statement, is also false. PHB degradation produces only carbon dioxide and water. No, that's not true. It can produce a variety of products. I also have three more questions, which I think I'll upload. Maybe I'll show the question in the video. I'll just quickly walk through the question. The substrate involved in co-metabolism are growth associated. The fact is whether it's true or false. So let me just explain it right away. It's false. The substrate involved in co-metabolism uh, are growth associated as well as the co-metabolic substrate. One substrate is growth associated maybe, but other is surely not growth associated. So this statement is false. If you remember what a co-metabolism is. Transformation of co-metabolic substrate produces toxicity. I guess by now you will all know that it's true, because the co-metabolic substrate will not help your microorganism to grow, and the substance that eventually we get from the transformation is going to be a toxic product for the cell because it's a pollutant. So here is the explanation, which is also present in the lecture slides. You can go through. So with that, we come to the end of week 7, live session 7. I hope you got some information from the session. So you can let me know if you want any changes or any additions or anything in the sessions. I'll just wait for a few seconds or 1 minute. Just let me know if the session is fine or any other suggestions for the session. Is the session fine, or should we change something with the way we go? We are almost past half. It's a 12-week course. We are in week 7. I'm just asking concerned with this live session. Any response? Yes, no? Okay, if you have any suggestions or any comments, you can always let me know through email or discussion forum, or maybe in the next session. That would be fine. Also, I hope all of you are able to access the um, recorded lectures in YouTube, as well as the slides content that is being uploaded in the drive. So for those who don't know, uh, after this session, I'll just upload the videos. Every week the videos is there in the YouTube. You can just go back and see if you want. And also I'll be uploading the PPTs as PDF in the drive. You can access it. I hope all of you are able to access. If you have some issues with it, you can let me know. So with that, thank you all for joining in, for interacting, and for your patient listening. I hope the session was somewhere useful to you. It was really great handling this session. Thank you all for joining. If you have any comments, you can leave in the chat box, or else you can leave the session. Thank you for joining in.